Hi everyone, welcome back. So, last time we talked about certain environmental conditions that can be very limiting for bacterial growth. Some species will struggle a lot, for example, with like a highly salty environment. That some species will be not well equipped to survive under those conditions. These are the non-halophiles in this example. Then you have species that have evolved to survive under those conditions. The halophiles in this situation that have developed certain strategies to help them live in an otherwise very challenging environment. For the non-halophiles, you're going to see that if they can grow in those environments, then their growth rate has slowed because it's hard for them to survive there. Whereas halophiles, being well evolved to survive under those conditions, they do just fine. But salt is also this really important resource too. It's not just very destructive. In small amounts, we need that salt. All living things need salt, mainly for their ions that are inside them, that form them. So these are really important for certain metabolic processes, certain processes that a cell has to do in order to survive, to do specific tasks. And without these ions, those tasks don't happen. So with enough salt, without enough salt, I should say, bacterial growth can actually stall. So if you have like a limit to the amount of salt, or it's rare enough that it's hard to find, bacteria can also suffer that way. So if it's not salty enough. And this is because salt is essential for bacterial growth. It's essential for growth of all living things to some degree. And we're going to explore a few different examples of some limiting resources like this. So bacteria ultimately need basically all the same things that humans and other living things need. A source of carbon, oxygen, nitrogen, and also other resources like phosphorus, magnesium, potassium, iodine, sulfur, and trace elements, things that they need in smaller quantities, like iron and zinc and manganese and things like that. All life, at its simplest, really requires the resources to build certain base organic molecules. We call these the biomolecules or biological molecules. You'll often see it written out. These are the basics to build our cellular structure, specific enzymes, DNA, sugars, like ATP and similar molecules, things like that. And so these are made of carbon, oxygen, nitrogen, hydrogen, sulfur, and phosphorus. Some things made of some of those things, others made of different ones. But these are like the basic, most essential elements of life. All life needs a lot of other resources too, other elements for more niche processes, more specific tasks, like those sodiums and potassiums and irons and manganese and things like that, in less quantity. But they're still very important. And they can definitely be certainly limiting if they're rare or not present. So we use these resources, for our basic biological molecules. And so these are the biological molecules, just to review. Lipids, first and foremost. These are what give the structure to our cell. It's what builds our plasma membrane. And a lot of cells also use these for signaling to one another. Certainly that's the case in eukaryotes like us. We, for example, in our body, we produce a lot of hormones, and a lot of our hormones are lipid-based. So, this is one thing that we use our lipids for. Carbohydrates are used for a diverse range of things, but one of the really important things that we use them for is a source of energy. Okay, so these are carbon rings that can be chained together, and also they can be broken apart and separated and used to fuel our respiration pathway, which is what we use to build ATP. So we can actually take the energy that is trapped inside of chemical bonds of this sugar molecule, and we can transfer that using respiration into ATP molecules for us to use under more specific circumstances. So, essentially, like, does any particular enzyme needs energy to do a specific job, we can just plug in an ATP molecule and use the energy trapped in that. So it's a bit like changing currencies, if you understand me. And so, this is also a source of energy. Our lipids also can be stores of energy, but carbohydrates are more immediately accessible forms of energy. But we can also use these for a bunch of other things. For a bacterial cell, for example, both the cell wall and also the glycocalyx, if it has one, those are all made of carbohydrates. Then you have the amino acids. Okay, these are the building blocks of proteins. So all of a cell's enzymes, all of their proteins. In the case of bacteria, also the little chains that hold the peptidoglycan, like the carbohydrates and peptidoglycan together, those are all made of amino acids. And so all of like the big jobs that a cell has to do, those are all made from proteins and enzymes, and therefore amino acids. Essentially all the things that a cell has to do, it's facilitated by these structures. So it's super important. Then you have the nucleotides. Okay, nucleotides are the backbone of DNA now, right? This is what builds the building blocks of DNA and contains the information inside of DNA. All right, so this is how we store all of our genetic information, is in the sequence of specific nucleotides, one after the other, that ultimately codes for something. It codes for the instructions on how to build a specific protein, for example. So there are a few other instances, a few other uses of nucleotides. For example, ATP also contains a nucleotide. Your ribosomes are also made of nucleotides. It's RNA instead of a proper enzyme. So there's a few other situations where you'll see those things for sure, and they'll have other major jobs. But this is definitely a really important, function, maybe the most important function of our nucleotides. Now, bacteria have to ultimately find all these different resources, all the stuff that we just covered, in order to continue growth. They have to either, get all the raw materials to build those things, to build those lipids, those carbohydrates, those, nucleic acids, and the amino acids, or they have to acquire those specific things themselves. Like they have to take up sugars, take up carbohydrates, or take up nucleotides, things like that. And some species are going to be more picky about what they need than others. A fastidious species is one that requires very specific resources or specific amounts of those resources. So, for example, a species that, say, can't digest, can't utilize like a bunch of different sugars like lactose and sucrose, but can use glucose and fructose. Well, that's what they need. They're a bit more fastidious than a species that could utilize all those different sugars. A non-fastidious species on the opposite end is one that is just less picky. It doesn't have as many specific needs, and it's more of a generalist. So going back to talking about culture media, well, a good culture medium is one that, bacterial growth is going to be supplied by these different resources. So we're essentially like providing the essentials for these different species. You will never find a universal growth medium, because there are some species that are fastidious, that require specific conditions that are not easily replicable by a specific medium. And also, you can even have situations where fastidious species can be so different from one another that they need virtually opposite things. Species that can only thrive in high salt conditions, for example, and then species that can only thrive in low salt conditions can't be grown on the same plate, because how would you do that? So that's a challenge for a culture medium. We're going to have culture media that is specific to the needs that we have for it. Now, many of these resources, these are things that we could consider limiting if they become scarce. So, what I mean by that is it can become a resource that not every bacterium can get access to. That they can be rare enough that some individuals may go their life without acquiring, while others do. And so, as a result, that can severely impact growth and reproduction, so that some bacteria may never actually be able to reproduce because they don't get that limited resource that was otherwise essential to reproduction. They might even die without it in some cases. So this is what we call a limiting factor for growth. So a good example of this is oxygen. Okay? All life requires oxygen, just not in every form. Elemental oxygen is present in every biological molecule. Okay? So this is something that is going to be universal. It occurs in all of the building blocks of life. But not all living things breathe atmospheric oxygen and get that oxygen that way. So some species do. The ones that do are what we would call aerobic. It means that they're capable of using oxygen in respiration. That while we make ATP, we take the oxygen out of the air and use it in order to take the energy out of a glucose molecule and shift it over to build ATP with it. On the other end of the spectrum, we have anaerobic life. So these are living things that are incapable of utilizing oxygen in respiration. Okay, the process, if they have, they all have a respiration pathway, they won't all be the same, and some of them will need it and some of them will not. The anaerobic species do not require that. There's also a lot of variation within anaerobic life. Living things are not also just like, oh, I can use oxygen or I cannot use oxygen. There is actually some variety here, meaning that oxygen can be a limiting factor in different ways for different species. So when we have terms like obligate and facultative that we want to cover here, because obligate means that you absolutely need this thing. You will not survive without this specific resource. A facultative species is one that, for whatever that resource is we're talking about, it can survive with it, it can survive without it. It may still have a preference. It may prefer to live with this resource or prefer without this resource, but it can live under both circumstances. Okay? So in the case of oxygen, an obligate anaerobe is a species that requires no oxygen, because if it is in the presence of oxygen, it will die. It cannot survive in an oxygen-rich environment, and it will have to be separated from that oxygen if it's going to survive. A facultative anaerobe is not that same way. It's more anaerobic out of convenience sometimes, where it's like these species may prefer to use oxygen, but it would be all right if it didn't have the oxygen. And so in the absence of oxygen, it can still survive and reproduce, but it might be at a slower rate. It might be less effective. You can also have aerotolerant species, aerotolerant anaerobes. These are obligate anaerobes that can still tolerate oxygen, but they can't use it. So they can live under some conditions of oxygen, but they really just never really utilize it. Okay? So there are some pretty interesting examples of this, and it's a bit different from what we typically see, because some species, most anaerobes have a pretty negative reaction to oxygen. And we can talk about that in a future chapter. But pretty significant. Then you have microaerophiles. Okay, microaerophiles, these are species that are they're aerobes. They need oxygen, but they require a very thin, like small amount of that oxygen. They need a lower concentration in order to survive. They do need it, but they can't survive in the high quantity that is typical in our atmosphere. This is a condition that is not typical. Like there are places where this occurs, but when we think about walking around outside and in the typical environments that we encounter, we don't see those environments that often. They definitely occur on Earth, though. So it may surprise you. Excuse me. It may surprise you to see that there's a lot of variation in oxygen requirements, right? Because Earth is really oxygen rich, but not all environments are actually as rich in oxygen as I just mentioned. Here's a great example in this video here of an environment, small as it is, where this cave has a very low oxygen environment, because it actually has this carbon dioxide sink that's being produced, where it will actually put out the torch here. It'll put out the fire because there's no oxygen to burn that torch. So the floor of this cave, again, a small environment by comparison, but not with respect to bacteria, has very low oxygen. And so there are some species that would thrive there because that's what they need, okay? So there's a lot of different examples of low oxygen environments, right? There's, some different cave systems that have very low oxygen. Places like the human gut, very low oxygen. Deep gas seeps, very low oxygen. There are places around the world where gases bubble up from the surface, from the soil. Also, like deep ocean environments in some cases, very deep layers of soil and aquatic sediment, and then even like small microscopic pockets can form of these environments where, again, bacteria, being microscopic themselves, can find a little home there and thrive for a good amount of time. What the? Okay, there we go. So oxygen is very troublesome, and the reason why is because it's quite toxic to most anaerobic life. Well, most life in general. It's kind of complicated, and we're going to explain that here, right? Even we are negatively impacted by oxygen, despite us really needing it. So oxygen is this really reactive molecule, all right? Oxygen gas. The main reason for this is because it can form what's called a superoxide, okay? This is essentially, in the environment, it can just pick up a free electron and become this superoxide molecule. When it's in this form, it's very reactive. It can, bump into and react with virtually anything that it encounters, at least in an organism. Like a lot of these organic molecules are prone to having a reaction with this chemical if it encounters it. And when you think about the different molecules that we have that build our cells, our lipids, our carbohydrates, our proteins, our DNA, if you just have reactions happening to these molecules, that can be pretty negative, because you can change the form of that molecule and therefore change its function. We can potentially destroy DNA, for example, by reacting it with a superoxide. This is what we call, you can call that sometimes a free radical. You can also call it a reactive oxygen species. So living things have a defense against this, okay? They have a defense to protect themselves from, from this detrimental oxygen. At least some of them do. The ones that have evolved in the presence of oxygen have gained this ability. Okay, and so we, for example, we living things that live with oxygen produce an enzyme called superoxide dismutase. So superoxide dismutase is super neat. It has this ability to react with superoxides and also with hydrogens. And essentially you smash them together and you make oxygen, normal oxygen, and then you make hydrogen peroxide, H2O2. Okay? That solves one problem, but it also creates another because hydrogen peroxide is also toxic. It's very problematic as well. And so other living things, some species, have devised another strategy to deal with that, which is that they have another enzyme called catalase. And catalase will bind to hydrogen peroxide and react it and eventually break it down into water. Okay? Just into water. So, big problem solved here. Okay? We're essentially taking the hydrogen peroxide, breaking it down to oxygen and water. But this metabolic activity, it's going to vary a lot across different species. Again, some species are not going to be able to do these reactions. They're not going to be able to do this this breakdown here. So in the presence of superoxide, species that don't make superoxide dismutase, they're going to just simply suffer the negative effects of being exposed to superoxide. Species that make superoxide dismutase but don't make catalase will solve this problem but not this one. And they'll be accidentally producing or unintentionally producing hydrogen peroxide. Okay? Now, superoxide for these species is just a constant problem. This is why anaerobic species cannot tolerate oxygen. Now, for these species, we can actually differentiate them using a different type of differential medium. So a great example of this is the fluid thioglycolate tubes. Okay, these are FT tubes. So essentially, what we do with these tubes is we prepare them in a specific way that allows us to give them a gradient of oxygen. So at the top of the tube, there's a lot of oxygen. A lot of oxygen dissolved in this medium. This is a broth, by the way. And then as you travel down through the tube, the oxygen quantity dissipates. It gets lower and lower until down here there's very low amounts of oxygen. Then you just stab the medium with your bacterium and you have it introduced throughout the medium here. And then you wait and see where it grows. Some species that are, for example, obligate aerobes, they will only grow at the top because that's where the highest amount of oxygen is. Other species will only grow at the bottom because that's the only place that they can survive, right? This is a good example of this right here. That this is like an obligate anaerobe where it avoids this higher oxygen quantity and only grows at the bottom. Then you have other species that will grow all throughout and they don't care. They just don't care, which places have high or low oxygen. And so this is an example of a facultative anaerobe. Okay, so we can actually differentiate between these species, between different life strategies using a medium like thioglycolate.